waves get lost in the ocean. 7 billion swimmers, man.
The last presentation for our lightning talk, we're going to ask Dr. Alastair Fortune to come back. Alastair this time is channeling a different person from the MS flagship. He's done a great job presenting his own work, channeling Kayleen. Now we're asking him to channel his colleague Roisin Maloney and present her work, How Can Our Genes Affect MS Severity? Thanks, Alastair.
Okay, so a bit of a different project this time. Looking at genetic mutations that might be impacting MS severity. So as we've heard a bit about today, the genetics interacts with our environment, and this is very important for whether someone gets MS, but it's also likely important of how fast the MS may progress. But the question is, how can we study this? So working alongside Dr. Nick Blackburn, who runs the MS Family Study, he's recruited a whole bunch of families with a high incidence of MS, so more than 3 first-degree relatives with MS, and did whole genome sequencing, so looking at their genetics, and was able to find, much like in his previous study, certain mutations that is associated with MS, at least in these families. And so to understand the changes that might be driving this, we translated this work and we started to look at how these mutations might impact the health of mice. So we put the certain mutations from this example family into a mouse to study how it impacts their health. And so over the course of a year, work I did in my PhD, I compared mice that had this mutation versus mice that didn't have the mutation. And so what we found was when we looked at the brains of these mice once they were a year old, so relatively old for a mouse, when we looked at a certain cell type called an astrocyte, which is a type of glial cell that helps support the neurons in the brain to work properly, we found that there were both more of them and they were what we call reactive. And so we know these cells, astrocytes in MS, are very responsive to damage. They release substances that can promote inflammation, but they can also release substances that dampen inflammation, so they're quite difficult to study. But overall in MS, it seems like they make it harder for nerve cells to repair after damage has occurred. So these variants that were identified in the family are impacting astrocytes within the brain. But this was under normal conditions, so the mice were perfectly healthy. The only thing that was different about them was that they had this mutation. And so we wanted to know if this mutation actually impacts the response to something like myelin damage. And so what Roisin has been doing is to use a model of MS-like injury. So this is basically giving them an immune stimulant, and it causes them to have myelin damage. So we can see how the cells are responding with the mutation to myelin damage. And so what she's nicely shown is that when you give this injury, the mice, the astrocytes, so this is in blue, GFAP is a marker of astrocytes, they are actually quite responsive. You get a lot more of them, they're brighter, they're covering more area of the brain. But specifically in the mice that carry the mutation and are given the myelin damage, there's this overreactive response. And so both at a baseline condition, but also once you've got myelin damage, these cells are responding too much. And this probably makes it much more difficult for the brain to repair itself. And so just thanking the team. Again, this is work from Kayleen Young's lab, and my PhD and Roisin's postdoc work. Thank you.
Thank you, Alastair. Across those lightning talks, we've seen the full gamut of our research from the MS Research Flagship.